What's really interesting now is that the factor IX gene that was used in the AMT-060 trial is a, let's say, conventional factor IX transgene. It's not hyperactive, it's just like a normal wild-type factor IX, even though the gene itself was codon optimized, but that doesn't change anything on the amino acid sequence of the therapeutic protein. Moving from AMT-060 to AMT-061, the only difference there is a point mutation in the factor IX gene. It's called a Padua point mutation, and it's an R338L mutation that actually hyperactivates factor IX. We and others have shown in preclinical models that this results in a 5 to 10-fold increase in activity based on the pioneering studies by Dr. Simeoni, who found this mutation in a thrombophilic patient. What's really intriguing now is that in the AMT-061 trial, and they reported now the first 3 patients that were treated with this vector, an increase in activity, which is exactly what you would expect using these hyperfunctional variants. So they're there's only 3 patients, so the statistics are not so robust yet, but it's hopeful that we are roaming in the range of 40 to 50% factor IX activity. This is pretty much in the same ballpark as in the trial that was reported in the New England Journal of Medicine of the SPK-9001 trial, where also about 30 to 40% mean factor IX activity levels were reported with the Padua transgene. Now, these 2 studies were not the first studies with the Padua. Let us not forget that Baxalta early on did report the first data on the use of a hyperactive factor IX Padua, where they saw a higher activity into 2 of the patients that were treated. But what is really interesting in these 2 trials, the AMT-061 and in the trial with the SPK vector, is that the responses between the different trial subjects were much more consistent than in the BAX335 study. We don't fully understand what the reason is for these different responses, but we believe that in part this could be related to the immune response. We do know that the T-cell immune response against AAV transduced cells can have a dramatic impact on the duration of expression. This has been published in the New England Journal of Medicine paper in 2017, where patients that have a high anti-AAV T-cell response actually have sometimes short-term expression of factor IX. So these cells are obviously cleared. This turns out not to be the case in all trials. There are some trials where the expression is sustained despite an anti-AAV T-cell response. And we believe that perhaps with some vectors we don't have an AAV-specific T-cell response at all, but maybe other types of immune responses. So we don't have all the bits and pieces of the puzzle together yet, but I think that's why it's important to have more trials to gather comprehensive data sets to get a better feel of what is going on here. The AMT-061 trial shows actually no unexpected safety issues. I think there is no evidence of, let's say, liver enzyme elevations. Maybe one patient shows a little bit higher levels, but it's still in the low end of let's say, in the high end of normal. One could barely speak about a serious adverse event here at all. In fact, it's very modest. And I think this is something we very often see in many of the gene therapy trials. If there is any transaminitis, it's usually at a very basal level. I think that's all very encouraging across the board. This is an acute phenomenon. If it does occur, it happens relatively early after vector administration. But one wouldn't expect any transaminitis, say, 1 year after vector administration. And this is exactly what we see in all these trials. If it does occur, it happens early on, and it doesn't happen again later. So this is really an acute effect. But even if it does occur, in some of the trials you can block it with, by transient immune suppression with corticosteroids. So I think there's a handle there that we can exploit to subdue this untoward immune reaction. As far as the AMT-061 trial is concerned, so the hyperactivity of the Padua is confirmed here. I think it's nice to see how these data compare to the AMT-060 trial, because here we can be sure the vector design is exactly the same. We're talking about the same capsids. So the only difference there is the effect of the Padua. And there the superiority of the Padua has indeed been shown. One of the difficulties in comparing the different trial outcomes is the fact that there are no standards. So it's a challenge really to compare what the outcome is of trial A with that of trial B because of the absence of standards. But nevertheless, we can have some idea about the overall efficacy and safety regardless of the absence of standards. I think the take-home message here is that the factor IX Padua is definitely better, and the safety issues are such that there's very encouraging in terms of vector safety across the board for all the trials that we have seen so far.